It took 3 billion years for life on Earth to evolve. Today, scientists can design and build their own unique living organisms in a matter of months. This film charts the journey of a group of young scientists who are learning how you go about engineering new life.
She's between them.
So what concentration do you need?
Oh, come on, Maddie.
Whatever she's comfortable with.
Can they cope with what is probably the biggest teller of whether you can be a scientist or not? Can you cope with consistent failure even if it's not your fault?
At one of London's top universities, a group of pioneering students are entering the IGEM Challenge, otherwise known as the International Genetically Engineered Machine Challenge. In just 10 weeks, the team has to design and build their own unique life form with a useful function. They will pitch their design against hundreds of competing teams from all over the world for the prestigious title of creating the best new life form. Creating synthetic life is like engineering a machine. Living cells are the hardware and DNA code is the software. To create their new life form, the team will be building their own DNA program and installing it into existing living cells. The new synthetic cells will take on the traits of the installed DNA, like a computer taking on new software. This remarkable new technology, known as synthetic biology, is a fusion between the design principles of engineering and our fundamental understanding of life.
So this is our base camp, and you should really make this your home. We're really glad that you've decided to join the team.
To be honest, I had no clue about synthetic biology. What was it? I didn't even know its name till we had a presentation after a lecture, and I just had nothing to do like for the rest of the day, so I stayed. So I got excited with the idea of getting involved into building a biological machine.
Not to put any pressure on you, but we've done extremely well actually in the previous years. The competition is getting tougher and tougher.
I like the idea of going and finding out something for yourself. You know, you proving something, you like providing all the evidence for this argument, and actually experimenting it, researching it yourself. It's amazing.
Hopefully we're going to give you a real insight into how one does research and science, etc.
Some of the supervisors came in my biology class and introduced us to the project, and I remember the first thing they said was, well, if you like weekends and if you like socializing and stuff like that, you know, don't bother applying. And I was, yeah, that's me. Well, anyway, I was fascinated by the idea of getting the knowledge that is already there in biology to a useful application. And that is what IGEM is about, I think.
So what you need to focus on at the moment, I mean, Chris will take you through this, but is starting to think of ideas for the project.
Basically, I am kind of a bridge between both the students and the advisors, helping communication to flow well between them, and also generally try to keep an eye on the team, try and look after them, and generally help them out, give them hints, advice, wherever I can.
This is going to be a hard summer for you. There's going to be a lot of work. But once you've been through the whole process, you will find it's a really, really exciting experience that you know, you really won't forget. And I mean in a positive sense.
So we started kind of brainstorming, coming up with ideas, and a few were dismissed quite soon, and a few, you know, we kept a few running at the same time.
The application, the thing is the application is really good because there is a demand to find out this kind of stuff, and there's a demand for a fast sensor, so when you combine the two...
The team is working fairly well most of the time in terms of coming up with what needs to be done and sort of allocating parts to each other. I mean, sort of, at first I tried to do more of organising, but now sort of other people are stepping up as well, which is good.
Do you think that is actually a realistic idea?
Yes. And it's quite possible.
I think a lot of people who go into like biology and medicine, they are often motivated by a personal background. And there's a disease in my family that I would like to tackle with my studies. It is a neuromuscular disease called Charcot-Marie-Tooth syndrome. Symptoms are that progressively your legs become weaker and weaker. My brother, my sister, and myself are affected by this disease. So that is an additional motivation for me to go into biology.
The team meet with their professors, engineer Richard Kipney and biologist Paul Fremont, to present their initial ideas.
Hello, my name is Nicholas, and you want to
Nick's come up with a complex idea to create synthetic bacteria that will communicate with each other to destroy pesticides in contaminated soil.
That will, if too many cells are killed.
For me, one of the really good things about IGEM is that the undergraduate teams that take part in this don't know what can't be done. Okay, so if you've got scientists like us, you know, we say, oh no, you can't do that, it doesn't work, etc. And they don't know that. And what happens is that really quite often some amazing things happen that, you know, you just look at open mouthed and you think, how did they do that?
And once the pollutant is eliminated,
it's come from them, it's all through them. And that's quite powerful, you know, in terms of their development as young scientists.
I'm Ben, and a few of us worked on looking at biological equivalents of logic circuits.
Ben's vision is to build bacteria that will act as the logic gates of an organic computer and harness the adaptive nature of living cells.
Which overall could effectively have an equivalent, a biological equivalent of a circuit that could rewire itself. Yeah, I think it's something that isn't at all possible in regular electronics.
Sure. I think the trouble with that approach, if you don't mind me saying, is that it can get very difficult. It's much better to have the application drive the technology. Yeah. That would be the critique on that. Is there any others?
Just quick.
Oh, quickie, yeah.
Okay, so we're going to talk about water sanitation. Quite recent work has been done on the autoinducer that...
Harriet presents an idea to create synthetic bacteria that will be used to quickly detect infectious diseases in water.
Another concern is whether or not it actually produces the autoinducer in water or only inside of us. Yeah, that's about it for now.
I think in principle it's a really well worthy of some really serious investigation, don't you think?
Yeah,
Does everyone agree?
Absolutely.
Yeah. What are we detecting then? All right, so
exactly.
Let's move ahead, right? So you've got it all working. What are we detecting?
I'd ideally like to be testing for parasites, but if we can't do that...
I think the concept of detecting parasites in water as an application is superb, and I think there's a real world-life need for that.
Yeah.
You know, it really is. It's still a massive problem. And they get very frustrated in the field about it because, you know, the problem is if they detected them before it got into the humans, it would be so much easier. You know?
Yeah, absolutely.
Everyone seems to be still smiling, which is a good sign.
It's going to be a bit of a roller coaster. You're going to find that in the middle you're going to think, oh, why did I ever do this? But I can tell you, no matter what happens, you're going to get a massive buzz out of this because it really is amazing, isn't it? Yeah. It really is. So think outside the box, you know, don't be constrained.
No, I agree. Well, at least for the next 3 days.
Right.
And then get firmly in the bloody box.
Now they have finalised an application, the team spends the next few weeks designing the DNA modules that they will need to run their synthetic bacteria.
Why do we have to put it last? Because we could use from the Crawl TV Telecom D as a master mix, basically, and then add in different...
The DNA modules will consist of the genes that code for the functional parts of their parasite detector, as well as other sequences that will control and support these genes.
The primers would add different sequences, right? They would add the specific...
Most of the DNA software will have to be extracted from living organisms by the students themselves. But the DNA that is hardest to obtain must be chemically synthesised from scratch, an expensive process that will be done by a commercial company.
Getting the sequences for the synthesis correct is absolutely vital, because if they find out that later on part of the sequence they have submitted to the company is wrong when it has been synthesized, we then have to try and work and repair whatever has happened, and that is a very, very difficult and challenging thing to do, and will add extra days and weeks to the time it takes them to get something working.
Cool, that looks good. Flanking sequence looks good.
Yeah.
So that's fine, that looks good. Looks good.
Having finalised the design of their DNA program, the team are ready to order the DNA that will be synthesised.
It's better if you send it. It's coming from you guys.
It's good. All done. Good. Woohoo.
So this is how we are going to create our synthetic life form. We're going to take existing living life forms, we're going to be using bacteria, and merge their DNA with the DNA that we have created. To do this, we're going to treat our bacteria with chemicals to make them take up our DNA. As soon as the DNA is on board the bacteria, we will have created our new synthetic life form, which will behave exactly in the way we want it to. The DNA inside our bacterium will behave just as new software. DNA in general codes for the functional parts of living organisms, the proteins. And the DNA inside our new synthetic organism will be processed to create the parts needed to give our bacterium the new designed function. The function that we have designed is to detect parasites in water. The particular parasite that we are going to be detecting is called schistosoma. It lives in water and causes disease in over 200 million people in the world, especially in sub-Saharan Africa. When we add some contaminated water to our bacteria, they will detect the parasite and change color. We have designed this color change response to be really quick, so you can just add some water to a tube of bacteria and quickly tell whether the water is safe to drink. Well, that's the theory at least. Designing the DNA to do that has been really complicated. But that bit's all done, so all we have to do now is see whether we can build our synthetic organism in the lab.
The first thing to do is prepare Petri dishes in which their synthetic bacteria will later live and grow.
You use this whenever you're using an amount more than a mil, or where you need something done over sterile. Alright. You need to take up 5.
Nick mixes antibiotics that will be used to control the growth of the synthetic bacteria.
Whoa. Do not want to flood the filter.
Go back.
Whoa. They're probably a bit nervous, probably a bit like, well, what's it going to be like? For Maddie and Nick sort of today, well, is this going to be the rest of the time spent with someone looking over my shoulder? Rather than, they'll be a bit confused as well, because they're learning new concepts and things they've not come across before, so it's a new environment, so they'll be a little bit out of their depth and a bit confused and panicky at the minute.
Okay, there you go. And just mix that.
That's a really cool thing.
It's back.
For me, was my first experience in actually being in a lab and doing actual scientific work, and I really like that. I really enjoy it. So I come here in the morning and I want to do stuff. The more I have to do, the more happy I am.
Don't worry.
Maddie pours a molten jelly containing the antibiotics into the Petri dishes.
There you go. I'll mix that for you. Just need to pour it. You need to be a little bit quick about this because this will start to set. There we go, that's good.
Doesn't matter how thick it is.
That's fine. Nick was very quick at picking up technical things so far, so that looks good. Maddie's always quite happy to put herself forward and actually give things a go, so that kind of spirit will mean they'll pick things up pretty quickly. Congratulations, you survived a day in the lab.
While the lab team gets started, other members of the group are still working away at computers. The bioengineers are building a website to present the team's work. And they're using computer programming to predict how their synthetic bacteria will behave once it's made.
the science aspect of sort of understanding the world around you. It's always been such an interesting kind of drive, just that curiosity about things. And then sort of the creative side, the wanting to make stuff. It's kind of engineering for me was just sort of the fusion between the two. Synthetic biology is about applying engineering principles to nature. We simplify all the complicated biology into parts and components. So, we call our host organism, in this case our bacteria, a chassis. And we think about our DNA as computer software. We think about the individual functional parts in terms of modules: an input module, a response module, and an output module. And when our synthetic DNA software is booted up, all our DNA modules create the functional components of the cell. So we have an input element, which is sent to the surface of the cell to detect the parasite. And we have a response element and an output element that both stay inside the cell. When the parasite is present, the input module detects it and sends a signal for the response element to be made. We have been trying to design this section so that it happens quickly, and we're calling this our fast response module. When the fast response element has been made, it activates the output module. Then, when we add a chemical to our system, it interacts with the output module to create our colour response. The beauty of thinking about things in engineering terms is that when the system has been built, the individual modules could be replaced by new ones. We could swap our colour output module for a sound module. Or we could swap our input module for modules that detect all sorts of things. Different parasites, specific diseases, poisonous gases, arsenic, or even gold. But in reality, the specific details of the system are really complicated, and that is why us engineers tend to spend lots of time modelling and remodelling the components.
In engineering, largely the problem is well defined. You know what the endpoint should be.
Yeah.
I mean, if you're trying to put a bridge across the Thames,
you kind of know what you're trying to do.
You sort of know whether the bridge is across the Thames or isn't, and whether it's working properly.
Indeed.
So yes, you have these problems in engineering, but you know at the end of it whether you've really solved them. And indeed that's what's really exciting about engineering, that there are these endpoints.
Yeah, I think that's what excites the biology community, because they can actually see an endpoint for their fundamental understanding. They can actually apply their knowledge in some way.
With preparations complete, the group starts building their 3 DNA modules.
Engineering biology is really, in many ways, not quite the same as doing it for normal engineering. Because of the tininess of the molecules of DNA, the things they're building, and the actual chassis, the bodies which will run them, it's almost a completely different ballpark. They're working with things they can't see, can't interact with so readily, and that adds a whole extra level of complexity to what they're doing.
No, it's fine. You only need one of those, right?
To carry out this formidable task, the group has split into teams.
It's now.
Yeah, it's part of the thing.
Yeah, and
kind of officially, I'm designated lab supervisor at the moment, and I'm trying to kind of keep an overview about all the different things that are going on, so that each of the different lab teams, they're very concentrated on what they're doing now. We have like 3 lab teams in the moment and running down 3 different lines of assembly. But like lab supervisor kind of implicates that you really have a clue what's going on, and sometimes I feel like I don't.
Oh my god, I'm actually really scared.
Harriet and Florian are constructing the input module that will create the parasite detection element. To achieve this, they must first extract a crucial piece of DNA from E. coli bacteria.
Kirill, can you pass me the lighty thingy from the other side? Now we're doing something practical. It's a lot less frustrating than when we were just sitting there with a laptop in front of us. It's also been really busy because there's so many of us in the lab. But I think that's nice. We can kind of talk in between, whatever we're doing.
To extract their DNA, they have split open their bacteria. The DNA is dissolved in the liquid, and they have to separate it from the rest of the fluffy white cell debris, a delicate process that mustn't be contaminated.
Yeah, I'm not supposed to move it much. I might put it back.
Florian prepares a filter that will trap the DNA when the liquid is pushed through.
Oh my god. I don't know how I do this.
Okay.
Oh yeah, lefty loosey. Okay.
Okay, put it in. And
Plunge it?
Yeah.
Gently.
Want me to?
Yeah, can you?
Whoa, shit.
Hold on.
No, this is not good.
I'm gonna go get Chris, hold on.
Okay.
I did many things wrong, I think.
Alright, I feel so sorry for you. I kind of
Just washed it left. Don't worry, weirder things have happened.
Maddie and Nick are working on the output module that will create the colour change element. While they wait for a crucial part of their DNA module to arrive from the synthesis company, Nick plans the steps ahead.
You have to really get on with your lab partner because you spend so much time together. And I know I'm really lucky, like I get on with Nick really, really well. And because of you've got to have like constant communication, you know, you need to really keep tabs on each other.
Make it 2.5 times bigger, that's 10, which is the final after we put all of this together. Just be easier, I think. Genius.
You need to have fun in lab as well, otherwise you just really will begin to hate what you're doing. But I'm having fun. I know he's having fun, he likes it. So I'm quite lucky. But I know other people, there's been tension in other groups.
2, 3.
Kirill is teamed up with Kiasha to build the fast response module.
Kiasha was always the quiet one, but now she's so starting. She doesn't mean to, I don't think, but she just sounds like she's kind of getting a little bossy, which is kind of funny. I mean, she's not really like saying, "Oh yeah, do that," because yeah, I'm telling you to. It's just yeah, the way she talks to people sometimes can sound a little bossy.
So I've done 1 and 2. You just need to do this.
What are you on about? We have to have 4.
Okay, yeah.
I think it's better that one person undertakes one thing so that they can keep track of it and, you know, just they know where it's going. If 2 people do it, it's kind of like you depend on the other person to see that you're doing it right, and you just tend to lose track of things.
How long do you need to spin for, though?
It doesn't matter. It's just on
Just put on. No, but it was balanced.
That's because you had a balancing now.
No, because I meant to text everyone saying party at Nick. Because I was texting you first, so like Wolf was in my head.
Oh, I've got something good for you. Guess what this is?
Synthesis.
Synthesis, yes. So now let's get into this first.
No, we don't have anything.
Sure. And then nice little set full of DNA. Wow. Do I open it up?
Yeah. Oh, that's an anticlimax.
Oh yeah.
Cool. I don't know what I was expecting when I opened that.
Today, actually, the synthesized DNA from an external company has arrived. So we now have these large pieces of DNA which they need in order to build their various constructs. Hopefully another one's been sent off today. Hopefully that one won't go via Italy. This one's been on holiday, by the way.
In biological research, we're trying to understand some of the fundamentals of how biological systems work, how natural systems work. And nature is a very difficult opponent, and one that I think is exceedingly difficult to defeat.
Maddie and Nick have used the synthesised DNA to build their output module. Kirill takes responsibility for the crucial step of getting the DNA module into their bacteria chassis, a process called transformation. He mixes chemically treated bacteria with the DNA. Next, he heats the mix up to shock the cells into taking up their new DNA software.
Whenever you're ready. The heat step is very, very crucial. If you leave it too long, the cells will start dying. If you don't give them long enough,
then they haven't been shocked quite enough to actually give them sort of a jolt and say, whoa, whoa, start doing something.
3, 2, 1,
go. It's kind of like getting somebody out of the bed in the morning just by giving them a little bit of a nudge rather than throwing a bucket of water over them. Then you try and keep them on ice for as little time as possible after that.
The bacterial cells are pipetted into nutrient-rich broth to recover. The bacteria are streaked onto the antibiotic jelly that Maddie and Nick prepared earlier. The antibiotics will prevent all but the transformed bacteria from growing.
What? We want it to be lower because that's...
She's making me nervous. Chris, I can't really see it. Oh, okay, I see it now.
That'll do.
Okay.
Shall I put the lid back on for now?
The bacteria are placed in an incubator to grow.
Over the next few weeks, things will start to get stressful, so they'll probably have some early big successes, but this will be probably quickly followed by a few major, almost crushing at first knockbacks where things just don't quite work and they start to begin to see and understand just actually how the majority of the science we do actually fails on a day-to-day basis. It's only overall it finally ultimately works. It'll be an interesting few weeks.
While the group wait for Nick and Maddie's transformed bacteria to grow, they meet with a panel of social and ethical experts. They discuss the wider implications of creating a synthetic life form for detecting schistosoma parasites in water.
Problems. 200 million people being affected by this disease obviously has a big impact, and even though it is mostly not lethal, it has a big impact on children, basically. Yeah.
I can see that it's an important disease, and it conjures up this image for me of this person who's living next to a water source that's contaminated. And if they discover that it's contaminated, what are they going to do? Because there might not be any other water sources.
A lot of the time, so they have an option, maybe so there's a lake really nearby that it's easy to kind of get water and bathe in and stuff. There's also a borehole that, or you have to pump water out through, and it's much easier to get the water from the lake. But if they know that there's, for example, schistosoma there, they would, take the effort to pump the water or go to another water source.
A bit of extra effort can be 6 hours walk.
For, say, a woman in her 80s. It's not necessarily an option. I mean, there seems to be an assumption sometimes that this water might be dirty, but there'll be other water that's clean. There may be people for whom there is no clean source of water.
Sure.
So you were saying, well, it's all dirty, don't use the dirty water. Well, then what?
There's nothing worse than white missionaries coming in to tell Africans what they need or what they don't need.
Try and let go of that West knows best, where come in and tell you what you need to know and what you need to do.
You're doing it for a particular reason. You're not just mucking about for no reason. Although even if you try and do something good with it, you get told that you're a naive person who just wants to save the world, which is really discouraging.
In this case, with schistosoma,
you can leave.
There's also the option of just leaving. So if you go to the lake and get a bucket of water out that you want to use for drinking or washing, and you use our detection mechanism and say the color is red, saying there are some parasites in it, a way to kill those parasites is just leaving the water for 24 hours, because the lifespan of these infective stages is only 24 hours. I think the main issue that troubles people maybe will be not so much the specifics of what exactly we're doing, but just the general concept that we're interfering with the natural course of things, whether that is creation or whether that is natural evolution, that we're interfering with that. And people feel, I think, that by doing this, we're taking a lot of responsibility on ourselves.
I want to know the worst possible application that you can think of where your product could be used.
Something that senses human skin and then attacks the cells and destroys them. Right. I mean, that's not what we want to do. But
no, of course it's not what you want to do, but it's
important to think about.
You're putting something out there that might help do things you have no idea about. So just for yourselves, between yourselves, just keep, thinking of the whole picture.
I think generally the idea of putting one gene from one organism into another, a lot of people are scared about that, including me. Someone puts a fish gene into my vegetables, how do I know it's safe? How do I know that it's something that hasn't changed the vegetable in a sort of profound way? How do we know that it only does what people expected it to do and not something completely different because it's a completely new organism?
I just don't think the public would let us get away with having a non-safe bacteria.
Yeah, but it's not getting away with that. You've fallen into the trap of getting away with. That's not the right language to be using. And I think you have to get out of that thinking, because synthetic biologists, we're not trying to get things through loopholes or hurdles. We're leading on these considerations. Can I just ask the engineer at the back there, what do you think? You're a bioengineer. Are you happy for me to go and release a lot of organisms out into your back garden? Or?
No, not particularly, but obviously
Exactly.
But some people that I've spoken to still do have the perception that it's just not right. We shouldn't be tampering with things.
Moral issue.
Yeah, it's kind of both put together.
Different debate.
Well, English was not my first language, so when I first heard the phrase playing God, I kind of wondered what it meant exactly. But ultimately, even bacteria genetically changed organisms. A good example is the agrobacterium. It naturally injects DNA into a plant to make it do something, that basically to feed it, which is essentially what humans have been doing for centuries. We've been genetically modifying wheat and things by selectively breeding them. It's not something we haven't done before, or it's not something that other organisms haven't done.
Gill, I don't know if you remember him. Was it Robin Gill? From Kent University. He's Professor of Theology there. And the question was asked to him about, does he agree with scientists, playing God and create? And he wasn't worried about it at all, because he said what God did is God created everything, and that we're just sort of playing around and fabricating, messing around with it. We're not actually creating, So in his religious context, creation is where the core of the belief comes from, and so therefore he's very cool about it.
You shouldn't shy away from those issues.
Don't shy away from the issues, but also don't, because of your assumptions. So
I think this is probably the first time, at least in biological sciences, that societal aspects are coming in and interfacing directly with the research.
In a way, that's groundbreaking, isn't it?
It is groundbreaking. I don't know of any other science field that's evolving in that way, and I think the social scientists and ethicists find that very exciting. Often what they do is they come in at the back end of a revolution, and they're kind of picking up the pieces and trying to say, oh dear, look at all this and what we should have done it better.
Yeah.
But at this point, they're actually in at the ground zero. So if we both get it wrong, we can blame each other.
Yes.
Back in the lab, Harriet and Florian have performed a chemical reaction on their extracted DNA to amplify it. They're using a specialised technique to visualise the DNA as white bands. However, they've noticed there's an extra band of DNA that shouldn't be there.
We need to try and work out where your contaminant is, to know what bit of your mix you need to throw away. It's your water, your primers, or your DNTPs. I don't know how you contaminated them. That's quite impressive. They don't know where the contaminations occurred. Unfortunately, we know it's quite a way back, but we don't know how far. So unfortunately, they're having to test for this now, which is for them an annoying step, which is costing days. You run one with the buffer changed, and you run one with the primers changed.
Okay, should we go and make those up now?
Yep.
Let's.
Ethan.
The transformed bacteria containing Maddie and Nick's output module have grown, and the group must now test them to see if the module works. But before they do this, they duplicate their bacteria into fresh Petri dishes, because testing the output module will kill the bacteria.
So when you take the stuff off...
The output enzyme exists in 4 parts, and it takes a while for a cell to make it from scratch. But we want our output to happen really quickly, so we have deliberately designed DNA that, in advance, will produce an inactive form of the output enzyme. The 4 parts of our inactive enzyme will have extra blocking proteins stuck to them that will stop the enzyme from coming together until it is needed. This means that our synthetic life form will be ready to spring into action as soon as a parasite is present. When a parasite is detected, the input and response modules will lead to the formation of an enzyme that will cleave the blocking proteins from the output parts. Then, when we add the chemical, the active enzyme will convert it into an orange-coloured compound. We don't want our genetically modified organism to be released into the environment, and one way to stop this would be to give it a death module. But a clever part of our design is that the colour-changing chemical will actually kill the bacteria, so it will kind of double up as a death module.
The duplicate bacteria are left to grow overnight, ready to test in the morning.
Tomorrow's going to be a big day. If nothing works tomorrow, then I don't know what we do then.
The chemical used to test their output module is a carcinogen called catechol.
This stuff is dangerous.
A numbered grid will allow identification of any bacterial colonies that change colour.
You've got them all?
They wait for the colour change that will reveal if they have correctly built their module.
Right, so this is the results of your catechol assay.
The assay didn't work. We didn't get the result we wanted. It didn't change colour.
So, okay, so you're expecting it to change from colour to something new. So, what do you think is the most likely reason for that?
You work really hard, like there's so many steps before you come to the testing stage. So you invest so much time, so many hours, days, or whatever. So when it goes wrong, you're just really upset, or it's frustrating, and I don't know, maybe you question your abilities, maybe you're doing something wrong, you feel like there's quite a lot of pressure.
Sometimes it doesn't happen. It's just the way it goes. Look at the lad. We've only tried it once, so if we give it a second go, see if it works a second time using a slightly different method.
Biology is so unpredictable, and especially you can't control a lot of things. A lot of things just happen out of pure chance or luck. That's what everyone always says. These things happen, these things happen. So, whatever.
This now needs to be bent, destroyed, removed permanently. Well, currently it's draining, it's hard. Sometimes you'll be very, very happy, sometimes you'll be very, very sad. About the closest thing I could ever describe it to being is having temporary bipolar disorder, as you're constantly either on top of the world or at the bottom of the well again. So, unfortunately, I'm now back on the coffee. My attempt to quit coffee for the summer has finally come apart. I was holding back the tide with tea for a while, but no, last 2 days I'm back on the coffee.
As experiments fail, the morale in the group begins to suffer.
Seriously, Chris, he's being really difficult. Like when he had some
people are difficult.
Give me another example other than today in the morning for the most difficult.
Oh, most days, trust me.
Oh, give me one. I'll let you hear.
What, do you want me to give you an exact date?
There's no point in doing this, guys. Neither are you going to get anything out of it, okay? People are difficult. Sometimes people just don't quite work well together, don't quite gel the same way. This can be worked around, it's not an issue, okay? Yes? Is that okay, Phil? You two are going to work separately, which is absolutely fine. I need to know which bits you're each going to work on.
Okay.
Carol? You have a say in this as well, Carol. It's not just Cass's choice, it's your choice as well. Do you have any preference, Carol, to what you prefer to be working on? It's your choice as well, Carol, it's not just hers.
Whatever she's comfortable with, she can do.
Yeah, you can sit inside this. Ultimately, you put people in this kind of high-pressure situation, you keep them locked up like this, it's going to happen. It's just very, very hard for people, so there's obviously going to be the occasional irritation, and unfortunately they've been having a particularly hard time lately. Things haven't been going quite their way, so unfortunately it's all boiled over on this occasion.
This allows them to really see, how a research lab really works. How things don't actually work out the first time. How you've got to,
they get brought down to earth quite quickly.
This is not easy. You've got to, you hit brick walls. You've got to think how you get around that brick wall. I mean, this is all the stuff of normal research that we're, this is our everyday lives.
You have to just believe that you think you can do something. That's you have to have an internal belief that you can actually achieve something doing what you're doing. Because if you don't have that, you shouldn't be doing this kind of game.
It's a new day, and Harriet and Florian are identifying the source of their contamination.
Primers. Oh, so it's that one that was contaminated, number 2.
That's beautiful. So it's appeared when you've changed the buffer.
2 is, the advanced buffer. 3 is the primers that we used in the first one. 4 is the water.
So I think the main thing there is you need to ditch that buffer. That's probably what's gone wrong. Some days it happens. Pretty much reality is, pretty much any lab reagent, no matter how careful you are, will contaminate given enough time. It's the lab equivalent of cancer, basically. It's just a matter of time until you get it. A bit morose, but it's true.
Maddie and Nick have rebuilt their output module and have again installed it into their bacteria. They're getting ready to repeat the colour change test. To get an idea of what the test should look like, the team first try it out on a strain of bacteria that contains the original unmodified colour change enzyme.
Yes, working. Okay, now ours.
Now the team try out the test on their own synthetic organism.
That one is.
Yeah, there's definitely a yellow one at the top. Don't touch it.
Yeah.
There's definitely yellow colour on number 7 and number 6.
Maybe just
Actually, 8 and 9 are going as well. 2 and 3. 14, 15, they're all going. All this plate now is going yellow. It means that there's definitely at least one positive on these plates.
They're all becoming.
Look at that.
Yeah.
Particularly, it's just where all the cells are. They're all going bright yellow. Big positive result.
I didn't expect it to work, and I was honestly amazed when we started seeing a colour change. When you achieve something, it's a really, really good feeling, so it makes it all worthwhile. Like I was thinking the other day, I'm really glad I'm doing IGEM, even though during the interview the professors were like, you work such long hours, you don't get a holiday. It made it sound really, really bad, but the actual work itself is really interesting. Like, there are moments when I'm just tired and exhausted and I want the weekend, but I feel like I'm getting loads from doing this lab.
A very good standardised test module as well, which is really good.
So occasionally, maybe once or twice in your whole life, you might actually understand something that nature has created in some ways.
And when you,
and that's about it.
If that happens on that rare occasion, I've had that happen to me a couple of times, and you sort of think, well, I may be the first person ever to have thought about this in this way and to have understood it, and that's amazing actually when you actually feel that.
The team have reached the end of the 10 weeks. They've designed a brand new synthetic life form for detecting parasites. They've achieved the major milestone of building the module that will show people whether their water is safe to use.
I think it's been really fun. I actually do, like especially the first few weeks when we were brainstorming and putting the plan together. And now looking back on our kind of whole system, it's amazing. I don't know if we'll get it done, but I think it's quite elegant.
It showed me that although it's hard, although it's competitive, although you need to be really focused, at the same time you're having a great time doing the actual science. So I grew really fond of science from the start of, it totally reshaped my opinion of being a scientist.
I initially was drawn to synthetic biology because of the nature of how fast you can put together something like a project required by IGEM. So we were already seeing results, and that's amazing. I know we're not quite there, like in regards to the entire system working.
The group have volunteered to work extra hours in the lab to try and complete the project.
I guess the main feeling right now is, how far will we get? I do want to finish everything, but that's not quite realistic. But hopefully we'll get to quite a good amount of data, which will be useful, because it'll show that we've actually done each bit individually, so we know that if we put them together, they should work, at least in theory.
In a month's time, the group will all travel to the IGEM finals at MIT in America to present their synthetic organism. They will battle it out with more than 100 competing groups from all over the world for the prestigious title of creating the best new life form.
Going to Boston, to MIT, one of probably the best university in the world, and meeting all these teams that have been working on the same project or the same kind of project for the whole summer, that will be an amazingly dynamic and full experience, I think.
Synthetic biology, I think all of us kind of hope it's going to become something big. Hopefully the field will take off. And I get to say, there at the beginning of it, right at the start. And I think that is that'd be quite a nice thing to have later on. And then if the field amounts to nothing and it doesn't really take off, then I just won't tell anyone.
The reason for me to switch from law into biology and science was this disease that my family suffers from. IGEM and synthetic biology gives you confidence in that manipulating life can actually be successful in this way. That gives you confidence that with a disease like that, you might even be able to tackle that properly. The principles you apply, the way you think, that has been an important lesson, and brings you forward. Absolutely.
Synthetic biology is an amazing tool. Once we harness that, you could do so many things, and why not try and save the world?
Everybody on quiz. There's something wrong with you, man. Why am I always praying for things? You made space for Wolf. Oh, so much pleasure.